Welcome everyone to this session of MedTech and Computational Bio talk. And today we have the pleasure to have Hari Shroff from NIH. And Hari's been a core developer of advanced imaging, especially in microscopy, for the last couple of long years, I think. A quick intro to Hari. Hari received his bachelor in bioengineering from the University of Washington in 2001, and under the supervision of Jan Lippard, did his PhD in biophysics at the University of California at Berkeley in 2006. And he spent 3 years as a postdoc with Eric Bexic at Geneva, where he focused a lot on palm microscopy, which is a difficult but very, very high resolution method to visualize molecules at the nanoscale. And he's been at the NIH for quite a few years right now, and heads the section of high resolution optical imaging and really developing the next cutting edge imaging tools for biological and clinical research. His talk today will primarily, I think today, focused on how modern computational methods are really driving imaging tools, and particularly in the aspect of restoration. I think one of the challenges in microscopy, apart from having just these beautiful images, is that we do face issues with phototoxicity and long-term imaging. So computational tools have really helped and advanced a lot of those techniques at the moment. And without any more introduction, I think Hari, please go ahead. Yeah.
Great. Thanks so much, Steve, for inviting me. It's a real pleasure to give an update on what we've been doing, and I really appreciate the invitation and your attention during this talk. So thank you. So as Steve said, I guess what gets me up in the morning is microscopy. So I'm an optical microscopist by training, and over the years, we've built tools that can image sort of single molecules and protein distributions within living cells. At somewhat larger length scales, the plasma membrane and cytoskeletal proteins in 3 dimensions over time. And then at even larger length scales, sort of the movement of cells within whole embryos, or at even larger length scales, whole organs that have been cleared and imaged with light sheet microscopy. And the sort of unifying theme in all of these examples is that in every case, we used computation to improve the raw data acquired by the microscope. And that's kind of the general theme that I'd like to speak about today. So in the first part of my talk, I just want to talk a bit about deconvolution, right? So this idea, of course, has been around for decades. And the idea is that your microscope never actually gives you the object. Instead, what you get is this blurry, noisy image of the object, which isn't the thing you actually want. But if you know the blurring function of the microscope, the so-called point spread function, and if you understand the noise process, deconvolution is this computational method that allows you to partially remove the degradation of the microscope, right? So you can kind of, at least to some extent, improve the effective contrast and resolution of the image. And like I said, this technique has been around for decades. Now, deconvolution can be used to improve the raw data acquired by a confocal or wide field microscope, but it also can be used in these more complex multiview microscopes. So some years ago, we invented this Dualview light sheet microscope, or DISPIM. And the idea being that, you can see in this image of a worm embryo with nuclei in the worm embryo, if you image it from just one view, then the axial resolution is always worse than the lateral resolution, because the objective lens collects fewer spatial frequencies along Z than in XY. And if you can come in with a second view, that you can actually compensate for that poor axial resolution by fusing the images, by registering one view on top of the other view and applying deconvolution. You can get a more isotropic image, as you can see on the right here, where the axial resolution is very similar to the lateral resolution. And this idea has been built upon by others. So Philip Keller at Genelia Farm has built a multi-view microscope useful for much larger specimens like fish or mouse embryos. And again, sort of the same idea, right, of fusing views, registering them, and then deconvolving them. And so in these methods, the main challenge is the computational burden. So you can acquire the data, the whole volume, in a fraction of a second, but it takes you a lot more time to register and deconvolve these views. And this was kind of a bottleneck in our research. And so we wanted to speed up registration and deconvolution. And that's what I want to talk about in the first part of my talk. So it's useful to kind of go back to basics and think about how deconvolution works. So, here I'm showing you kind of a classic deconvolution algorithm, an iterative deconvolution algorithm invented by the astronomers Richardson and Lucy back in the day in the 70s. But this method is still very powerful and it's still used today by us and by many others. And you can think about this deconvolution method as moving between two spaces. The space of the object, this is the thing you actually want, and the space of the image, this is the thing you get with your microscope. And Richardson-Lucy models this process as moving between the object of the image using some forwards projector or forwards function F, and then backwards from the image back to the object using some backwards projector B. And this simple convolutional equation, which you can code in a few lines of code, describes how to improve your estimate E of the image I by convolving it with this forward function F and convolving again with this backward projection B. So the forward function F, or the forward projector F, capital F, models the physics of the microscope. This is like a point spread function. The backwards projector B, well, you have some flexibility in picking that function, but typically it's taken to be the transpose of the forward projector. And so what this iterative equation here says, if you do this backwards and forwards mapping enough times, say K times, eventually you can improve your underlying estimate of the image E. And so if you can reduce the number of iterations, K, right, the number of times you go backwards and forwards between the object space and the image space, this can directly translate into faster deconvolution. And so, the natural question is, if you want to speed up deconvolution, can you reduce the number of iterations, right? The number of iterations K. And so we started thinking about this, and like most ideas we've had, there's nothing new under the sun. So if you go back a few years in literature, we found this nice paper in Nature Methods by Prybish and coworkers, where they showed they could get this reduction in time by choosing a different back projector. So instead of using the transpose of the point spread function F hat, if instead they made this point spread function a bit more Gaussian, they could reduce the computational time. And this strategy is called choosing an unmatched back projector. So we read this and we wondered, how much speed improvement is possible in principle? And again, if you go back even further in the literature, you find out that really there's nothing new under the sun. So some radiologists back in 2000 showed this intriguing result that you get faster Richardson-Lucy if the eigenvalue spectrum of this product of projectors F times B clusters together, or is as flat as possible. Okay, so what does this actually mean in terms of fluorescence microscopy? Well, in fluorescence microscopy, you can write down your imaging equation as a convolution, right? The convolution of the object E with the point spread function F plus some noise term. This gives you the image. And when you convolve in the spatial domain, you kind of mix spatial coordinates. However, if you Fourier transform this equation, this convolution becomes a product of terms, and now you nicely kind of diagonalize this product in these spatial frequencies. So you get this nice equation, this product of Fourier transforms. And another way of interpreting this equation is you can say that F and B are diagonalized by the Fourier transform. And so the eigenvalues are given by the Fourier transforms of F and B. So this now lets us link this imaging equation back to this first statement that I gave you. And so the punchline is, if you pick B such that the Fourier transform of F times the Fourier transform of B tends towards a constant, or 1 if you normalize, or if you rearrange these terms, if you pick B such that its Fourier transform tends towards 1 over the Fourier transform of F, or in real space, if you pick B such that the convolution of F and B tends towards a delta function, this guarantees a faster deconvolution. And for those of you that think about transfer functions, this is picking B such that its Fourier transform is like an inverse optical transfer function. So, again, this is kind of abstract, but this is a prescription for improving the speed of iterative deconvolution just by changing one term, B, in this iterative equation. You can't mess with F because F models the blurring of the microscope, but you're free to pick B so as to change Richardson-Lucy deconvolution. So here are a couple of examples of how this actually works. So suppose you image, some structure here, mitochondria, imaged with a high-speed structured illumination microscope called the Instant Sim or iSim. This was developed in my lab some years ago. And if you look at the raw data acquired by this microscope, it's okay, but it's a little bit blurry. And the blurriness you can characterize with the point spread function F, either in real space, or you can talk about its Fourier transform, right, in frequency space. If you use Richardson-Lucy deconvolution, using the traditional back projector B, which is just the transpose of F, you can sharpen this image. And that's what I'm showing you in this bottom row over here, that with 15 iterations of traditional Richardson-Lucy, you can get a resolution limited result. But it takes 15 iterations. And that we can understand this by looking at the product of F and B in Fourier space. It's not particularly flat, right, up until the resolution limit. So it takes some number of iterations. But if we start to mess with this back projector, this B, if we start to tweak it so that we start to amplify the high spatial frequencies, we make it, say, more Gaussian. Now it takes us only 9 iterations. And if we look in frequency space, the product of F and B is a bit more flat. And you can sort of take this further. You can further amplify B such that its product with F in frequency space is more flat. Now it takes only 4 iterations. And if you derive a kind of limiting form, this so-called Wiener-Butterworth form, now it takes only 1 iteration to get the same resolution limited answer to within noise. So just by changing one term, by changing B, we've reduced the number of iterations a factor of 15, and we've sped up our deconvolution. So again, the punchline is that we've tweaked B such that we've met this condition up till the resolution limit, and this gives us a faster deconvolution. And this works across many different microscope types and samples. So you can image microtubules in a confocal microscope. You again see this kind of progression in speed as you tweak this backwards projector. You can image, say, nuclei in a light sheet microscope, a single view light sheet microscope, or you can image actin in a wide field microscope. And in all of these cases, there's about an order of magnitude speed up simply by changing one term in this deconvolution. So, you know, the outputs of the deconvolution are the same to within noise as the traditional deconvolved result. And this also is helpful in these more complex multi-view imaging systems. So going back to this dual view light sheet microscope, if we look at a concrete example here of worm embryos, where we label the nuclei in magenta and the neurons in green, you can see in the raw data, you have this kind of anisotropic data where the axial resolution is worse than the lateral resolution. You can deconvolve that data using traditional Richardson-Lucy deconvolution, and you get a more isotropic result, but it takes 10 iterations. Using this Wiener-Butterworth result, we get the same result to within noise. We've sped up our deconvolution by a factor of 10, right, in speed by changing this one term in the deconvolution. And one can imagine even more complex microscopes. So if you look at a T cell, an immune cell, where we've put in actin, a GFP marker of the actin, and imaged it in this triple view microscope, where we acquire actually 4 views, triple objective system, where we acquire 4 views of the specimen. Now the raw data is kind of blurry. We can improve the resolution and contrast using deconvolution, but it takes us 90 iterations. And using this Wiener-Butterworth trick, we get this factor of around 18 improvement in speed by changing one term in the deconvolution. So this works. The point is, this works in more complex deconvolution schemes as well. Now there's a problem if you try and use these multi-view imaging specimens, in that you have to register the data before you deconvolve it. And what we found is that the registration is more of a bottleneck than the deconvolution. So by registration, I simply mean take the volume from one view and map it onto the other view before you deconvolve. And the method that we use for doing this registration is an image intensity-based method, where we're basically searching for the affine matrix that kind of, best cross-correlates, if you like, one image volume onto the other. That's very general because it just cares about the image content in each volume, in each view, but it's traditionally slow to do that search. How slow? Well, if you look at the C. elegans embryo or the T cell data set that I just showed you, you can see from these graphs, it takes a lot more time to register the data than to deconvolve. And if you look at even larger data sets, in this example here of a posterior lateral line primordium, this is a group of cells that is migrating in the living zebrafish embryo that actually forms neurons. This is a 2 terabyte data set. A couple of teravoxel data set. Here, it would take sort of an insane amount of time to register the data, right? More than 1,000 hours using classical methods. So we realized we also had to speed up the registration as well as the deconvolution. And we were able to do that. So there were kind of 2 tricks. One trick is simply to port everything onto a graphics card. So you can make use of parallelization on a GPU card if you rewrite the registration algorithm in CUDA. That's fairly straightforward, right? But the other thing we did is that we improved the underlying registration method itself. So instead of trying to kind of solve for this 12 degree of freedom affine matrix, we actually could break down this search and first optimize translation, then the rotation degrees of freedom, then the scaling, and then the shearing. And by doing this kind of sequentially, we got both a more accurate registration and an increase in speed. And so this method of doing it is 10 to 70 times faster than competing methods that are out there for kind of registering data. And we use this not just for registering multi-view light sheet microscopy data sets, but also for doing stitching on very large data sets. So going back to the zebrafish example, the reason I think this is important to keep thinking about, right, to improve the post-processing of the data, is that in this example, we could acquire volumes in 30 seconds. And we were acquiring many of these volumes, right, over 7.5 hours till we acquired this large data set. Any one volume could fit on a graphics card, which meant that using these methods that I mentioned, we can actually keep up more or less with the speed of data acquisition. And so this was important because what the biologist actually cared about were the shapes of these cells. And so he had a simple segmentation routine he could use on the raw data. If you try and apply the segmentation on the raw data, you get just about half the cells. If you use these deconvolved data sets, you get almost all of them, right, compared to the ground truth. And so this was very useful to him to be able to kind of process the data quickly. And you could even imagine if you could further improve accuracy and speed of feeding back, right, and applying perturbations to the system with some pulsed laser or with some optogenetic perturbation. And so this ability to kind of interact with the sample in real time, which we didn't quite hit here, I think is an important application which kind of should motivate us to keep improving the speed of these post-processing methods. Another application that I think is worth highlighting is when you're trying to image just very, very large data sets. So with these clearing methods like iDisco and Clarity, and with better objective lenses, it's become possible to image really vast, really large amounts of sample that have been optically cleared so that your light doesn't scatter very much as you go through it. The problem here is that the data set could be terabytes or even tens of terabytes or petabytes. So to try and register and deconvolve these data sets, even in this coronal brain slice that I show here, this wasn't so large. It was about a terabyte in multiple colors. It would have taken us hundreds of hours to register and deconvolve the data that it would take us like 10 minutes to image. And so there's this mismatch between the post-processing and the data acquisition, which is just a bottleneck, right? So this is another reason why I think it makes sense to keep speeding up the registration and deconvolution in this case. On a consumer grade graphics card, we could drive that time down to a couple of hours. And with more GPUs or with better GPUs, you could further improve that speed. So this is just another example case, which I want to flag, which is kind of increasingly important, right, in biomedical imaging, this ability to kind of generate these isotropic data sets where the microscope acquisition is not the bottleneck, the post-processing is the bottleneck. One final example I want to talk about. So, it's always bothered me that in all of these microscopes we use, they're hopelessly inefficient, right? I mean, in the case of this Dualview light sheet microscope, we work at moderate NA. And so we collect less than half the fluorescence right off the bat, not to mention all of the optical losses in the imaging system. So it would be nice in some applications to collect more light at once. And one way of doing that is to, instead of putting your sample on glass cover slips, put them on reflective cover slips, a mirror if you like. And so this mirror has the function of reflecting the light sheet back. So now you have two real light sheets instead of just one. This means you can collect more data per unit of time. But it also has another advantage, which is just like when you look at yourself in the mirror this morning, say, you get a view of yourself that you may or may not want to see, but it's certainly a view that you don't normally see unless you look at a mirror. That also works in microscopy, right? Because if you image something on a mirror, you get these virtual views of the specimen because the light that would otherwise be lost through the cover slip has a chance to reflect back and be collected. And so you can increase the collection efficiency in this particular example 2 to 4-fold, depending on how you count. The trade-off is that there is this increase in epifluorescence contamination because each objective lens doesn't just see the clean light sheet. It also sees this out of focus light. And you can kind of see that at right here in the raw data, right? If you look at the raw data, you can see that you get these 4 views of the specimen at once, but you also see this out of focus haze. Now you can model that haze, it's kind of well behaved, and you can remove it using deconvolution. The problem is that the point spread function varies across the field of view. And so you no longer can make this assumption that the point spread function is the same everywhere across the field of view. And so now again, deconvolution becomes a dominant computational burden. It takes, instead of just a few convolutions per iteration, it could take hundreds or thousands of 3D convolutions per iteration. When you consider tens of iterations per time point, this means that for the C. elegans embryo expressing GCamp, this example here that I'm showing you, it would take like 14 minutes to deconvolve one volume that you could acquire in a third of a second. So again, there is this mismatch between the computation and the acquisition. And using this Wiener-Butterworth trick that I mentioned, you can drive that down an order of magnitude, but it's still 90 seconds. So it still takes a lot more time to deconvolve than to acquire the data. And so we wanted to also try and speed up deconvolution in this case, this difficult spatially varying deconvolution case. And so we turned to deep learning to do this. Now, deep learning has been getting a lot of buzz lately, but of course the idea goes back to the 50s, right? Where you had these perceptrons, and the idea is if you have your data and some feature you want to learn about, if there is a statistical mapping between the data and the output, the thing you want, stacking these nonlinear filters together and using gradient descent and backpropagation gives you a very efficient way of learning this mapping. And this has been used, for example, in segmentation or in prediction, where if you have bright field images of cells and fluorescent images of some organelle in the cell, if you train these neural networks with these pairs of images, then given a bright field image, these networks are shockingly good at returning the nuclei, for example. And so we wanted to make use of this same idea for deconvolution. So we trained a neural network we called dense deconnet to take in these 4 blurry images, these 4 views of the specimen, and then given ground truth data we got the hard way, using either the Wiener-Butterworth deconvolution or the classical Richardson-Lucy, we could train the neural network to retrieve an image that resembles the ground truth. And this works very well and is very fast to apply after the network is trained. So here's an example looking at GCamp muscle transients in these worm embryos. You can see the raw data, which you can sort of see what's going on, but it's contaminated by this haze. You can see the traditional deconvolution, the Wiener-Butterworth deconvolution, and then the deep learning result. And the deep learning result is sort of almost to the level of Poisson noise in the images, right? So you really would have a hard time distinguishing the deep learning result from these more traditional results. But best of all, it really drives down the computational time so that now with the trained network, we're much more on par with the speed of taking the data. And this actually makes it feasible to use this microscope, right? The ability to sort of keep up with the computation also lets one play games with the microscopy. So you could use, for example, a lattice light sheet microscope with a really high NA. And so we sent some of these cover slips, these reflective cover slips, to our collaborators and who had this lattice light sheet microscope. The idea being that with the lattice light sheet now, in addition to the high NA primary view, the mirror gets you this virtual view at no additional cost. So you get a second high NA view from a different angle, which gives you spatial frequencies not captured by the base microscope. And so if you could fuse these data and deconvolve it, you can actually improve the axial resolution of the base lattice light sheet microscope. You can almost double it, so you get a more isotropic reconstruction. But it would have been unfeasible to process this data conventionally. With the Wiener-Butterworth trick, you get this 15-fold improvement in speed. And with deep learning, you get another 160-fold improvement in speed. So if you use the trained network, it again becomes possible to process this data in almost real time. So to kind of summarize on this part of my talk, I've told you a few things. There's this trick you can make use of in deconvolution where you change one term and significantly reduce the number of iterants. And that can be useful in different kinds of microscopes. I've told you about ways of using GPUs to improve registration, and then also using deep learning to further improve speed, which I think could be useful in these difficult to deconvolve cases where the kernel, the point spread function varies across the field of view. And there are many folks that are using this code or some variant of it. You can email us if you'd like to try it, or just try it yourself. All of the code is open source on this GitHub website. And I've just kind of given you a preview. So please read our paper if you'd like to dig in more, and then email us. So I now want to talk in the second part of my talk a bit more about deep learning and sort of perils and pitfalls with deep learning. So the motivation here is that it's very hard to build microscopes that have awesome spatial resolution, are really fast, don't fry or sample, and have superb signal to noise ratio, right? I mean, if you optimize one axis, then it comes at the expense of other axes. And this makes sense, right? I mean, you can't have your cake and eat it too with physics. with optical physics anyway. But using prior information and neural networks, you can extend the space of what's possible. You can make a prediction that lets you in some sense beat the trade-off in the microscope. And so, there have been a number of papers that have, provided very compelling illustrations of using deep learning. And so we were interested in these techniques. And we were particularly interested in characterizing the limitations of these techniques. So I want to talk a bit about that right now. So the workflow in these methods is typically the same. So here I'm showing you what would be called a supervised workflow for denoising, where you have some target you're interested in, you turn your laser down and you take a low SNR image of that target or a volume, then you turn the laser up and take a high SNR image of that same target. You acquire a number of these matched pairs, you train your neural network, and then given a trained neural network, you challenge it with new noisy data the network has never seen before, and then you ask how well does the network denoise the data. And so, the particular neural network that we favor in our lab is this particular thing that we didn't invent called a residual channel attention network. This attracted us because the authors claimed that it was better at preserving high resolution information than alternative neural networks. And this is sort of very attractive to us fluorescence microscopists because we always care about resolution. So if we can get denoising without compromising resolution, that's great. We collaborated with a small company called S-Vision, which is now part of Leica, to extend this framework to three-dimensional fluorescence microscopy volumes. And I'll talk a bit about our study, which you can read about. It was published recently in Nature Methods. And again, the code is available open source on this website if you'd like to try it yourself. But let's look at sort of some data. So if you image, say, mitochondria, with an instant SIM, this high-speed structured illumination microscope I mentioned before. Here is an example of the raw data fed into the network. It's very noisy. This particular target labels the outer membrane of the mitochondria. So when we image it, we have some expectation, if we image it high enough resolution, that the mitochondria should appear hollow because the fluorophore goes to the outside of the mitochondria. And you can see that in the raw data, you can sort of see it in the XY, but in the axial view sucks. It's terrible. You can't see much of anything. It just looks like haze. If you turn the laser intensity up 100-fold, now the ground truth-like data, you can actually see a lot, right? You can see both in the XY and the axial view that this fluorophore goes to the outside. The tag goes to the outside of the mitochondria. And so if you train with enough of these pairs, you can train different neural networks, the RCAN, the CARE, which is a UNet, or at the time, state-of-the-art neural networks, SSRSNet and ESRGAN, and you can ask how well do they predict the ground truth. And what you see here, both visually and quantitatively, is that all of these networks offer a bump over the raw data, right? They all do a much better job compared to this raw data input. All of them resemble the ground truth. And one can quantify how well you do using these metrics like structural similarity or peak signal to noise ratio. These are global metrics that sort of ask how well do you recover the ground truth. All of these networks do a good job. The RCAN has a slight edge compared to these other networks, but it's small. And this kind of performance was recapitulated in many other organelles we looked at, like lysosomes and microtubules, actin, Golgi, and endoplasmic reticulum. So in these single cells, in this kind of 100-fold power regime, we do fairly well with these neural networks. And I think the real value of these denoising neural networks, of course, is to apply networks trained on fixed cells to live cells. So to just give you an example of this, if you look at mitochondria imaged in this instant SIM, the instant SIM is not a light sheet microscope. So we can't maintain this high SNR for very long before we just bleach the sample. And you can kind of see that here in this movie, right? Within 10 volumes, as quantified in this curve, we extinguish the fluorescence budget. The images are no longer useful after 10 volumes. One way of beating this trade-off, of course, is to turn the laser down. If you turn the laser down to the point where the rate of photobleaching is small to the rate of protein synthesis, that's amazing. You can image forever. The problem is that the signal to noise ratio is garbage and you can't make much use of those images. And you can kind of see that in this movie that I'm playing now in the right, that in the right-hand movie, we can image for a hell of a long time, but the images are pretty poor quality. Using the RCAN trained on fixed cells, we were able to recover the data to the point where we could actually get super resolution images that were in principle segmentable. And in this case, we imaged for a few thousand volumes or tens of thousands of images without any detectable photo bleaching or photo damage. In fact, the fluorescence actually increased over time, which is remarkable. I mean, at least for me as a sort of microscopist that has spent too long using these techniques, I mean, I've never seen this ever in a super resolution microscope. So I at least find this amazing, that effectively we can operate a structured illumination microscope like a light sheet microscope. And this scales to other kinds of targets as well, right? So you can look at, for example, lysosomes and mitochondria. Again, on the left, the raw data, we've turned the laser down so that we can image for several hundred volumes without obvious photo bleaching. But in the red channel, in particular, you really can't see much of anything. In the RCAN reconstruction, the result is sufficiently good. You can actually see and segment mitochondria and lysosomes and even quantify how these organelles interact with one another. So it's like night and day compared to the raw input and the RCAN prediction. So again, this is kind of very encouraging. This, of course, begs the question, where does the network fail, right? I mean, that was the next question, because of course, if you put in suitably low SNR data, it must fail. And so this was sort of an attempt to characterize that. If we look at the endoplasmic reticulum at really low SNR, and then look at the RCAN prediction on the left and compare that to the ground truth on the right, you can see that, well, one, sort of globally, the RCAN gets it right, but you've obviously lost some spatial resolution. There's a price you're paying. But there are also, if you look carefully, there are regions where the RCAN, it doesn't just get it wrong, it either misses information or it makes something up. And this would be awfully nice to be able to predict, right, when you apply these neural networks. So you know when can you trust the network and when should you be wary. This is a big area of research in this field. So one way of attacking this is to try and train multiple networks, all with different parameters, right? So we did that here. And then you can compare the mean network output to some measure of variance, maybe the standard deviation of the network output, or a more sophisticated statistical test, something called the Ensemble disagreement. The problem is that these measures of variance, if you train different networks, at least in our view, are not especially informative. I mean, we couldn't, the standard deviation and the ensemble disagreement were not especially informative as far as telling us where the network failed. What was much more informative is a simple signal to noise ratio metric where we converted the raw data into this signal to noise ratio. So given some assumptions about the number of photons in each pixel and the read noise and of the camera, we can make this per pixel SNR map. And what we found is that if the SNR fell to less than some critical level in the input data, the network gave us garbage. And so, sort of just giving you a few empirical examples here, if you zoom into the ER and look at these cases on the left where the RCAN gets it wrong, the ensemble disagreement or the sigma over the mean, the so-called coefficient of variance, I mean, they're not especially informative. You can look at these and then they're not predictive of where the network fails. If the SNR falls to below some level, this is where the network starts to fail. So this is very crude, what I'm telling you. And I think this is a plea for more research into this area. I think this would be an area where there could be lots of room for impact if we come up as a community with better metrics as to how well these networks fail or don't fail or give you good results. Okay, I want to move on to a different application now, which is resolution enhancement. So here the idea is you have low resolution data and you have high resolution data you somehow take of the same object. You use these pairs to train a neural network, and then you ask, given some blurry image the network has never seen before, how well does the network de-blur the image? And so we can play these games using simulations. And so that's what we started with. If you take point particles that are about the same size or a little bit bigger than the point spread function, and then you blur it a factor with a kernel twofold larger than the point spread function, you can then ask how well do these neural networks give you a result that resembles the ground truth. And what we find is that if you blur with a kernel that is twofold larger than the point spread function, all of these neural networks do a fairly good job, right? I mean, the CARE and the RCAN might be a bit better than the ESRGAN and the SSRSNet, but broadly speaking, they all do a pretty good job. You give it this blurry input over here in the upper left, and then they all give you an output that is pretty close to the result. The beauty of a simulation is you can now turn the blur up. So if you blur the data with a kernel threefold larger than the point spread function, now you start to see the network starting to be degraded, right? The ESR GAN and the SSRS net, especially in 3D, kind of fall off the rails, but even in the care, you start to see some discrepancies, and in the RCAN as well, if you look closely. And if you turn up the blur even more, so now you're blurring with a kernel 4-fold larger than the point spread function, now all of the networks show significant deviation. So in some sense, with these toy simulations, we can bracket where the network works and where it doesn't work. And so to just sort of summarize that, you know, the RCAN does a bit better than these other networks on these toy data for these synthetic noise-free phantoms. The performance degrades as you turn the blur up, so that, you know, the structural similarity indices and the PSNR drops fairly dramatically as you increase the blur, and there's obvious visual deterioration of all networks at this 4-fold blurring level. Nevertheless, these results on simulations were kind of encouraging enough that we wanted to try and look into real data. And so we had a collaborator that had this nice commercial STED microscope. STED is one of these techniques that won a share of the Nobel Prize invented by Stefan Hell. And the nice thing about the STED microscope is you can operate it in super resolution mode and also confocal mode. And so we imaged microtubules and nucleopore complexes and DNA using a DNA stain at different phases of the cell cycle. And again, we trained networks separately for each of these organelles, and we asked how well does the RCAN recover the STED image. And I think it's fair to say that in all of these cases, the RCAN is better than the confocal image, right? So it's both sharper and it denoises the data. So all of that is good. And I would say it approaches the STED data. So the level of resolution improvement is similar, but there are these discrepancies if you look closely. So in all cases, in my view, it beats the confocal input, but there are some discrepancies compared to the ground truth. And so this was, you know, again, sort of enough, it gave us enough confidence to try and apply these networks to live cells. So, for example, this DNA stain, we could train a network that looked at data at many different phases of the cell cycle, and then we could actually image cells using a confocal microscope and then recover resolution and signal-to-noise ratio. So it turns out that even with confocal microscopes, it's hard to get cells to divide at high resolution. But if you turn the laser down and operate the method in so-called resonant confocal mode, you can reliably get these cells to divide. You can see that image on the left. These are volumes over time. The cells can divide without significant photobleaching, but the input data that we feed the network is both very noisy, as you can see, and the resolution is at best diffraction limited. The RCAN prediction is quite a bit better. It's denoised and the resolution is improved, particularly as the cells, these cells over here resume their kind of interphase structure, right? There's all this detail you just don't see in the raw data. So this was kind of encouraging to us. We also wondered if we could train RCANs to improve the instant SIM, that's high-speed structural illumination microscope that I mentioned before. The problem in the instant SIM is we didn't have a means on the microscope of improving resolution. So we didn't have a STED that we could use to get improved resolution ground truth. And so we turned to this other method, expansion microscopy, which you may have heard of. This was developed by Ed Boyden a couple of years ago. And I think of this as kind of a copy and paste. So you take your fluorescent label, you copy it over to a hydrogel, you add water, the thing swells more or less isotropically. And then because the whole sample, or I should say the dye, has been swollen, then you can read up the data with a confocal microscope at higher resolution or an instant SIM. And so here's an example on the right of the mitochondrial and microtubule network read out with a microscope in our lab. And you can see the resolution is obviously higher than what you would get in your confocal. The major challenge we found here was the registration of the post-expansion data to the pre-instant SIM data. Even though globally there was this isotropic expansion, if you look real closely at the length scale of an organelle, there are local distortions. And this is the dirty secret in the expansion microscopy field, that on some length scale, there's kind of an anisotropic expansion. And this is really, really hard to get right. So we actually failed. We couldn't register the ground truth high resolution data to the instant SIM pre-expansion data. So instead, we got around this by using synthetic data. We took our post-expansion data and we blurred it and we added noise so that blurry and noisy data resembled the data that we would acquire raw in our instant SIM. So we have these two pairs of data, the post-expansion ground truth data, which we deconvolved, and we degraded that data by adding blurriness and noise until it resembled the instant SIM. And then we asked, to what extent does the RCAN improve real data acquired in the microscope? And so it does to some extent. So if you look at the, in this case, synthetic input data, compare that to the expansion data, there is some improvement in the Arcan, but it's not very much laterally. It is significant axially, and there's some lateral resolution improvement you get, but you don't quite do as well as the expansion microscopy ratio, which was a factor of 4. And so if you quantify that, in this example, a little bit less than 2-fold resolution laterally, you go from 170 nanometer laterally to about 90 nanometer with the Arcan prediction, but that's not as good as the expansion microscopy ground truth of 60 nanometers. Axially, we do better. We get around a 3.5 improvement in Z. And so these gains were still nevertheless encouraging enough to try and train neural networks for live cells. Again, I think that's where the real power of these neural networks could be, right? Because there are good ways of getting higher resolution data fixed. And so just to show some examples here, if we look at GERCAT T cells, these immune cells that are spreading on an antibody-coded cover slip, the ISIM input data after deconvolution is okay, but particularly in Z, we get this bump in resolution after the Arcan network. And if you look at these mitochondria, if we zoom into this area over here and look at the axial views, again, you get this kind of improvement in Z compared to the Arcan, the instant SIM input. So, I've given you some examples where I hope, you can sort of see how these tools might be useful, right? If you can build up training data on commercial systems. I now want to talk a bit about using this in conjunction with a non-commercial system, a very new system we've just built. But the motivation biologically is the following. So I'm kind of obsessed with the problem of trying to follow nuclei in the worm embryo. I would love to make the cell map of how all of these cells make the animal, even after the muscles form inside the C. elegans embryo. And so we invented this Dualview microscope to do this. And so, we can image C. elegans embryos with all of the nuclei labeled. We can get these embryos to hatch. The data look okay until I pause this movie, taken with this Dualview light sheet microscope. And if I pause it, especially in late frames, you can see that there are areas where we can sort of imagine segmenting the cells, but there are also areas where the images are pretty bad, right? Because of scattering and aberrations, it becomes very difficult to actually track these cells. So even though the microscope in principle has enough resolution, in practice, the sample defeats us because of motion blur, depth-dependent aberrations, and scattering. You really need diffraction limited resolution or better in all 3 dimensions. You need to be reasonably fast, speed better than a second per volume, and you can't tolerate any degree of phototoxicity, right? Because we're trying to understand what happens in vivo. And so we can still make progress on this problem by breaking the problem up into a set of simpler problems. Instead of trying to follow all the nuclei at once, which makes the imaging really hard, we can try and follow just a few subsets of nuclei at once, right? So if we target GFP to subsets of cells and then image them with the dye spin, now because the data are sparse, we can unwrap the data, we can segment and track it, and we can build this kind of composite model of embryogenesis. So I think this will eventually carry us over the finish line doing it this way, but it would be awfully nice to have better microscopes, right? So you could image more cells at once in a single embryo. That's the goal. Okay, so in order to do that, we built the following microscope, which is this triple view confocal microscope. So we come in with different objective lenses around the sample, which could be an embryo or it could be a cell or a piece of tissue. We collaborated with ASI to build these compact line scanning illuminators. These produce sharp diffraction limited illumination over several hundred micron field of view. So we take data from all 3 views, and then we register the data and deconvolve it, much as I told you about in the first part of my talk. And then the main benefit of doing this for small samples is this bump in axial resolution, right? So you fuse these 3 views and you can get 250-ish nanometer lateral resolution and close to 300 nanometer axial resolution with these 3 objective lenses. But the other benefit is that in thicker and more scattering samples, you can sort of compensate for scattering from just one side of the sample by getting better light from the other sides of the sample. It's unlikely the scattering is the same from all 3 views. And so this could be useful, for example, in tissue, even if you don't have tissue that is that thick. So in this case, this is a kidney slice where we labeled in 4 colors. You can kind of see the glomerulus, these distal tubules, and then yellow is marking blood vessels. If you image this from the side, the axial view along the XY view, what you see is from just one objective near the cover slip, you can get a lot of detail. As you go further and further in, you get this kind of smush because of scattering, because the resolution is degraded by this kind of scattered haze. If you come in from multiple views and fuse the data, you can collect data all the way through the sample. So for thick or even not that thick but scattering samples, this is an additional advantage you get with multi-view fusion, and this scales to somewhat thicker samples as well. So in this example, this is a whole fixed C. elegans embryo where we did label all the nuclei, and we think we got them all. So we used another deep learning method to kind of segment the nuclei. And unlike any point scanning or light sheet microscope, we could actually get sufficiently good quality in this whole volume to be able to segment all of the nuclei. So this was a good proof of concept, I guess, that in fixed, brightly labeled specimens, we could get this advantage over high NA light sheet or confocal microscopy. Okay, but confocal microscopes have trade-offs, right? Because you're illuminating the entire volume of the sample. And especially for kind of thickish samples, like this is a fairly large example, cardiomyocyte, which is 17 microns thick. These are the cells that beat inside your heart and make it beat. Here we stained it with mitochondrial dye. If you look at the raw data from any one of these live volumes, you can see that this triple view reconstruction is quite a bit sharper than the raw data from the high NA view. That we can compensate for the scattering. The Z resolution is better. But if we image this over time, not only is there some motion blur because of the need to fuse these three views as the sample beats, you can also see that there's this pronounced bleaching of the sample, right? And that's because we're bathing the sample with light all the way through the volume. So we need methods of extending imaging duration, imaging faster, and using less illumination. And this is where I think deep learning can really help. So we can use these methods I told you about before to kind of compensate for these failures in the microscope. So how did we do that? Well, in the case of the worm embryo, which I'm going to use as an example, if you look at the raw data acquired from each of these three views, you get a high sort of resolution view from the side that is nearest the objective in each of these views, but you get this kind of gradual loss in image quality due to scattering as you go in. You can fuse those data sets together and improve that, but at the signal to noise ratio you need to keep the embryo alive, the data are kind of noisy. So we wondered if we could use denoising first to denoise each of these three views. If you do that and then deconvolve the data, you can get a higher SNR reconstruction. But then we went one step further. We trained a second neural network to take the raw low SNR view and go directly to the final high SNR triple view prediction. So we use this kind of two-step neural network prediction. First, to denoise, and second, if you like, to fuse the data and to descatter. And the punchline is this works fairly well. By stacking these neural networks, you can get a high SNR triple view fusion using only one low SNR view. And that's kind of shown here. So if you look at the raw input data from just one confocal view, you can see the data quality is sort of okay from one side. It gets progressively worse on the other side. The deep learning prediction is much better. We can recover many of these nuclei on the far side, and we can still get the embryos to hatch. So this is sort of like a bit like having your cake and eat it too. You get your long-term imaging, you get fairly high quality imaging, and you get it at a speed high enough to segment more nuclei. Now, one of the advantages of the C. elegans model system is you know how many nuclei there should be, because John Sulston back in the day won a Nobel Prize for segmenting the nuclei and lineaging them. So this is in black here, the kind of curve. This is what we want to get. In red, you can see a segmentation using another neural network for helping us do the segmentation. But the point is, if you look at the raw data input in to the network, you get just over half the nuclei. Using the DISPIM, a state-of-the-art light sheet microscope, you do a lot better. But this triple view prediction gets you even more of the nuclei. And there's still some performance gap because of aberrations. So we're working on trying to narrow that. But this was, for us, a very encouraging result, right? That by using deep learning in conjunction with the microscope, you can do better than the base microscope in this task that we care about, right? Namely, segmenting nuclei. Okay, so one other nice thing, this feature of this multi-view line scanning confocal platform, is you can do super resolution imaging. So by blanking the line as you scan the line, by turning it on and off at high speed, you can collect a series of phase shifted images that are like a structured illumination microscopy image. Because you're isolating the fluorescence in each line, you can do sim post-processing to improve resolution along the direction you scan the line. right? And you can do this in 3 directions because you're scanning from 3 orthogonal axes. So this means that using these NA lenses, you can get sub 200 nanometer resolution laterally and 250 nanometer resolution axially. And of course, if you use higher NA lenses, you could further beat this down. The trade-off in this mode of operating the microscope is you need more images for the sim mode. You need 5 images per view, or 15 images compared to the classic confocal microscope. But we have developed methods that use the 1D sim ground truth and deep learning to predict the 2D sim output in each view from a single confocal scan. And so that lets us circumvent the limitation of collecting more data given the deep learning network. And just to give you an example, some examples as to how this works, you can take a confocal scan, say from the lower view, in this case of immunolabeled microtubules, and then you can apply the neural network and bam, you can improve the resolution. So you pick up this 2-fold improvement in resolution given only a single confocal input image. And you can imagine doing this from 3 sides, right? If you apply these 3 neural networks, now in live cells, you can go, for example, in a living cell with actin, the raw confocal image on the left is okay. You can see these filopodia, you see this rearward actin flow, you see stress fibers, but the multi-view confocal sim result is much better, right? I mean, it's a super resolution result with better contrast, and we get this with less dose than a classic structured illumination microscope because of this neural network, right? Because we've already trained it to predict the resolution in each of these directions. And because we built this on a confocal platform, this scales to thicker specimens as well. So we can look, for example, at the nerve ring in a live worm where we've targeted GFP to the nerve ring and some neurons. You can see the raw confocal input gives you some optical sectioning, but it's diffraction limited. The triple view confocal sim result is much better, right? There's just a lot more detail you can see in this nerve ring region in the dorsal and ventral nerve cords and so on. This is again faster and clearer, I would argue, than a classic structured illumination microscope. And you can read about this on bioRxiv and hopefully before too long in press. So, see, how am I doing for time? Do I have enough time to like just give a few more slides?
Yeah, a few more slides will be, yeah.
Okay, so I'll just give a few more slides. So one final example, which is kind of unpublished. So getting back to light sheet microscopy, which we use a lot, we routinely image these densely packed samples, right, where we have these nuclei that are packed inside a C. elegans embryo. And you can see as we go through the stack that on the far side, eventually you just get a smush, a haze, because of aberrations, because of poor axial resolution, even at fairly high NA, where you should be able to resolve these nuclei. It becomes very difficult to run segmentation algorithms in this raw data. So we've now trained ARCANs to kind of systematically dissect the problems. So we can train one ARCAN to remove aberrations, another ARCAN to improve axial resolution, and then using expansion microscopy ground truth, we can get super resolution data. And so by stacking these ARCANs in series, we can take advantage of the native strength of light sheet microscopy, the ability to go for long times and fast, but get much higher resolution. So both within the nuclei and to separate nuclei, we can get much clearer images using the deep learning prediction. And this is kind of exciting for me because we can again use the strength of the light sheet microscope to image at high speed to get many, many volumes, but then we can clean it up using deep learning. And I think this could be useful to kind of be a gentler way of attaining super resolution. So, you can use the images on the left to get these long-term volumetric time series, and then you can clear up the data in these regions where there's scatter. To sort of summarize all what I've told you in this last part of my talk, I think, the current paradigm is you have your microscope, you use it to take data. And if you understand the imaging model, you can clean this up a bit. But fundamentally, it's back to the drawing board if you need higher resolution data or faster data or more long-term data. I think the promise of deep learning is you can use the data itself to improve subsequent data, right? So you can sort of manipulate the workflow in a way that lets you bypass using totally different microscopes. And that could be very powerful, I think, for us, for our community. But of course, there are challenges as well as opportunities. And the number one challenge to solve is how much of what we see can we believe? This is fundamentally an ill-posed problem, right? So there are a family of solutions, and exactly how much you want to believe the solution probably depends on the task at hand. I mean, from the technical perspective, the question is how much can spatial resolution or signal to noise ratio be improved? And I've kind of touched upon that from like a microscopy perspective, right? An empirical perspective. But it would be great to have better theories that can better predict this for a given task. Of course, the other practical task is how much or how little training data might you need. As an instrument developer, I'm really interested in these hybrid workflows, right, where you build better microscopes and you utilize neural networks to kind of, inject more into the image. And of course, maybe this is science fiction, but at the end of the day, the biologist doesn't actually care about the image. This is the hard truth of our field, right? They want something in the image, whether it's a segmentation or a fluctuating calcium signal. That's the thing you actually end up spending time to do. So the question is, can you build workflows that just bypass the image entirely and just goes from the data to the thing you want? I mean, that I think is where maybe we're heading. But before we get there, there's a lot of work to be done, including just training, right? And getting these methods in the hands of biologists. So I think there's a big opportunity, I should say, not just a challenge in this field. And that's my view anyway. So with that, I hopefully didn't give you the sense that I did all this myself. I really rely on lots of collaborators at the NIH and outside, and particularly in my lab. I'd like to thank Yi Chong and Min and Xiao Fei for some of the instrumentation, and then Ryan and Pluto and Gigi for some of the biology help. So thank you very much.
Thank you, Hari. That's a fantastic work. I have no idea you went so deep into deep learning. That's really amazing. And I think my first comment would be, I think it's great that somebody finds some use for expansion microscopy as a ground truth. I think that was quite innovative. I think I've not really thought about that using as a relatively reasonable ground truth. So there's a few questions I have. So I'll just get quite a lot, actually. I don't know if I can cover it in this half an hour or more.
Please, yeah.
I think Tianan's got a question first, which goes back to the earlier part of your talk on reflected light sheet. Can the auto-focused reflected image be used to resolve optically using a remote focusing module in the detection path? So as I understand, right, using the refocusing module in the detection path to look at the out of focus reflected light. Yeah.
You could, yeah. I mean, there's probably more information in that out of focus light than we used, right? So for us, it's kind of a nuisance, but of course it does carry information. And one thing that I think would be interesting, like an interesting direction, would be to try and scan rapidly, right, using your refocusing and use that somehow. So that's interesting. I'd have to think more about it, but thank you for the question.
Right. I've got quite a lot, but I think I'll go
please.
Four. I think in the beginning of the talk, you talked about using Wiener as a way to reduce the decon process, and then you spoke about using a ML approach to train and speed up. But have you considered the training time, and have you used trained data?
It's a good question. So one other issue with these networks is that they're brittle and they take time to train. So using the best hardware today, right, for many of these things, it would take a day. A day would be sort of like on the short side to train, maybe more likely a few days, right? And so that is a limitation. And there's the fact that we generally train one network per organelle. Although if the organelles look kind of similar, maybe you could train a more general purpose network. I sense that both of these things will get better in the future. I think GPUs seem to show no sign of slowing down, whether it's parallel GPUs, right, or cloud computing, or just better single GPUs. I don't think we're near the limit there. And so I think advances in hardware, just basic hardware on the computing side, I mean, nothing that, we do, but just NVIDIA, for example, right, will drive innovations there. Now, whether you get to the point of real-time processing and real-time training, that may still be like a lofty goal, right? I'm not sure that one could get there, but maybe one could use transfer learning to drastically shorten the training time. So that's a good point. There is this investment in time upfront with deep learning, right, where you train the network, and that's much slower than actually applying the network. Applying the network could be fast, like seconds fast per volume.
Right. I guess the second one would be, you seem to have sort of trained for denoising and deep blurring. So it seems to suggest to me that they're very different problems, even though it's sort of the training data set might be the same. So I guess, long story short, if you look at that noise prediction, seems to be a clear research problem, right? So in some ways, I think prediction of noise is not well thought out as compared to deep blurring. So am I right to think that that's what you're suggesting?
Yeah, I mean, I think of them as two separate things, but I think that boundary is imaginary in my mind, right? I mean, I think that there is a sliding scale that one can use to describe images in terms of spatial frequencies and noise. And those two things are related, obviously, right, by the transfer function of the microscope and the signal to noise ratio in your image. So you're right that I've been thinking of them like experimentally as distinct things, but probably the whole thing ultimately comes down to noise, and we need better ways of predicting the noise. Yeah, so I've described, and I guess there is one thing I'd say though, which is that I found empirically that you do better if you can break the task up into distinct networks. And that maybe, I don't have a good explanation for that, except that it may just be easier. You've got some complicated loss function, right, where you're basically trying to find the minimum. It could just be that if you break this up and you first denoise, you're in a better place, right? You approach the kind of valley better than if you just try and do it all at once. It could be something as simple as that when you're training and applying these networks, right? Maybe that's why in practice you do better if you first denoise and then deblur. We found empirically, I didn't really mention this in my talk, in this triple view confocal, right? I gave the example of a two-step network. And we find that if we do this 2 steps, we actually do better than if we try and go directly from one view to the final high SNR view. And I don't know if that's for some fundamental reason or just for some stupid reason, right? Like the training isn't well thought enough. I don't know. I just don't know.
Okay, I guess the last one would be, I guess you actually done 3D sim using 3 objectives, and I guess there's a lot of questions there. How do you beat registration, syncing? Because this is not trivial to do in 3 objectives. Because even with one, it takes time to get the right face. So
yeah. So I can talk a bit about that. I mean, so the thing we did in this microscope is that we synchronize the line to the rolling shutter of the camera, right? And that is done in light sheet mode routinely by light sheet microscopy. So we did this with a diffraction limited line. So we synchronize the line. That's how we get confocality without having to descan and rescan. So we synchronize the line with the rolling shutter. So then your question is, okay, how do you do sim? So if you grant me that you can synchronize that line in all 3 objectives, then to get the sim, we use our AOTF to just turn on and off the laser rapidly. That's how we get the sim. And then by controlling the phase of how you turn the laser on and off, you can phase shift the signal. Does that make sense? I mean, that's how we do it.
Yes and no, but because it's going to be not isotropic, right? Because you're coming on very different, and
you have to do this.
Right?
You have to do this from 3 sides. And so we do this serially. We do this serially from 3 sides. We first take one side, then the other, then the other. There's no attempt to do this at once, which could be a cool direction. One thing we mentioned in the discussion of our paper is that if you could do this simultaneously or more cleverly, right, than just doing it one view, then the other view, then the other view, there could be potential for improvement in speed. We didn't go there.
I see. That sort of makes sense. Yeah, that's good. Yeah. Any questions from the audience? Feel free to unmute and ask, guys. Yeah.
Hello, I have one question.
Yeah,
from
please.
Alex. So I have a 2 questions actually. The first one is related to the deconvolution.
Yeah.
So just in the end, you end up come up with this winner Butterworth way.
Yep.
Just do deconvolution in one single step. How is that different from just directly do deconvolution once you know the PSF?
Yeah, so it's a great question. So I think the question, if I can rephrase, is there are single step things like a Wiener filter, right, where you can use. Basically what we found, and we didn't investigate this like in detail, but we looked into it a bit. We found that the noise was suppressed in our method versus the Wiener 1 step. So if you use a Wiener filter, then you tend to amplify noise, or it's very tricky that you have to really tune the parameters so you don't suppress the noise. Richardson-Lucy, that family in general, does a better job with noise because unlike the Wiener, there's some attempt to account for the Poisson noise, right? The Wiener filter doesn't account for the Poisson noise. It's appropriate for Gaussian noise, but our images really aren't contaminated with Gaussian noise, right? It's some combination of Poisson and Gaussian. So I guess the short answer to your question is that I think our result gives you a better image. The noise characteristics are better controlled with this than the Wiener, straightforward Wiener thing where you just divide by the PSF. Does that answer your question?
Yes, yes. I have a quick second question. It's about the machine learning. So once you train the data, train the algorithms, so that there's assumption that this distortion of the data is the same applied everywhere else in a sample, is assuming that there's like spatial irreverent across the sample. This is kind of the same as the convolution process. I mean, is this so when you deconvolute or guessing the data? Does this become a problem where you making that assumption?
I think the so in the case of the spatially varying point spread function, I don't think our network is learning a spatially invariant PSF, because I don't think there's anything in the network that teaches it that. It's only looking at these local patches. I think all it's doing is just sharpening the image. You know, I think what it sees are lots of examples of fairly blurry data, or I should say data with lots of out of focus light. I don't think it's doing anything fancy. This is something that would be cool though, right? If you had a network that had the ability to kind of distinguish, right, between larger patches and smaller patches in a clever way, then it might be able to learn that. But I think in our case, all it's learning is just a sharpening. That's my view.
Right. Okay. Yeah. Thank you.
Sure.
Thanks. Anyone else?
Yeah, I guess I had two questions. One was a little bit, it's more again related to the RL deconvolution at the beginning. So the effect of changing some of those like back projectors to get those different results. I guess each of those, it's quite subtle sometimes, the change in a back projector, which does give you a different result, right? So I guess, have you tried looking around like different samples and different, runs, how I guess I'm just wondering, if you compare it to a ground truth, what exactly is the effective, if you're getting a different result as the output, does it actually match like a ground truth? Or is there a particular ground truth that works? Or how do you deal with, again, I guess it's more about how do you evaluate the performance of it?
There might be very slight variations in the result, but I haven't looked super carefully at this. I mean, what I can tell you is that the difference, at least for all the stuff we looked at, the difference is negligible. I mean, between the classic Richardson-Lucy result and then this. I mean, one of the kind of unsettling things about any of these iterative methods is that nobody has come up with a way of saying this is a good stopping criteria, right? So for any of these methods, they can run off the rails if you deconvolve them. So I don't have an analytical proof that says that our method will converge to the same result. But I would argue that, even if you have such a proof, you actually don't want the result to converge, because the result of any of these ML methods that converge is just garbage. It just over deconvolves, right? The maximum likelihood result, is terribly noise amplifying, right? So I'm not really rigorously answering your question, except to tell you that I think if you actually looked at the difference, you would have a hard time telling the difference, even quantitatively.
Right. Okay. Yeah. And the second thing was about, so I think so when you're chaining those ML networks, I guess.
Yeah.
Sorry, I mean, when you're training some of your deep learning methods, was it? Yeah. So I guess what happens when, we were previously talking about how it's difficult with even one run of the network to get to make sure that it is close to ground truth, or that is in itself an exercise. If you're chaining multiple of those together, I mean, is there something more complex that would happen, or is it fine to do?
It's a good question. I mean, what you're basically doing is you have some ground truth, and the network is trying to minimize the difference, right? There's some loss function that it's minimizing. I mean, so I guess I don't know the answer to your question. I mean, I haven't looked deeply at the difference between these multiple networks. The only thing I can say is that loss is lower, in the final result, right? So you have some final thing, which might be an expansion microscopy image or something, right, of an embryo, because you're trying to minimize. And if you do this multiple step thing, the final thing is closer to the ground truth than if you just use one. But but yeah, I mean, this is alchemy, man. I mean, like, I wish there was something more rigorous.
Yeah.
The whole field is kind of like the Wild West right now, you know. It's just a problem.
Yeah.
It's no problem. I mean, yeah.
Okay, cool. Thank you.
Great. I think it's been a fantastic journey you took us from
Good.
simple deconvolution down to the most extreme. I think.
Yeah.
That's yep. That's it. Thanks everyone for being on the call.